Hey everyone, if you can hear me, I just want to let you know that the meeting will begin at 7. I started the meeting at 6:30 to give us a little bit of time to make sure we're all squared away. So I apologize if there was some confusion. In the future, I'll start them at 7 and just try to join a little bit earlier. I'm still trying to figure out Zoom, so my apologies.
Stuart, what's new in West Seattle?
Mute. West Seattle.
Yes.
The large flowered colomias and the varied leaf colomias have germinated and are starting to grow.
Okay.
And how about you over there in wherever you are, Donna?
I'm in Wedgwood. Well, I don't have any exciting nature news to report, except for today I did walk behind at the Montlake Phil, saw some ducks. I saw ducks and including beautiful wood ducks, and I saw turtles sunning on a log with their legs extended out. I guess they were trying to get maximum sunshine.
Could be.
Yeah. And Carolyn, anything special going on over in Fremont?
No.
Any cabbage whites?
What was that?
Any cabbage whites?
Oh, I haven't been outside that much, but I usually see them every day. I mean, so, but yeah, no, it's been raining too much. And yeah, I probably haven't seen them for a week or more.
Richard, you will.
Hi,
Go ahead.
just checking in, getting a voice check. This is Mr. Pelham.
Oh, hello, Mr. Jonathan Pelham. How are you? Welcome.
Well, thank you very much. I saw a slug today,
Whoa.
a native slug.
Okay, that's good.
I did, you know. I live in like the worst butterfly habitat in the world. But I do want to say one thing. I introduced a colony of purplish coppers to my my apartment complex. You know, we have a lake here, and we get all kinds of them, you know, bony winged things. You know, what do you call them again? Oh, birds. And so I brought in some females last year and turned them loose because there was a bunch of the polygonum, the wetland polygonum species. I don't know which one, Stuart. Can't remember the names, but there's
There are a
about 3
few
or 4.
wetland polygonums, but the first one that comes to mind, polygonum, oh, it's escaping me.
Yeah, I know. Viviparm isn't it? I can't. Persicaria?
Well, Persicaria is it's not so much a wetland species. They're not necessarily.
Well, anyway, so this June, you know, I didn't see them, and through July I didn't see them. That this August they showed up. So we have a butterfly species in Snohomish County, or at least one I can verify.
That's pretty cool.
Yeah, it wasn't bad. I think it's illegal to import butterflies for the purposes of establishing colonies, so don't tell on me.
Okay.
Yeah, it's asking me to show my video, but I can't because I'm naked.
Oh my. Well, I have an infestation of moths in my pantry. So I constantly find little larvae and I throw them in my compost bucket. So I'm hoping eventually to persuade them to move out of my pantry, but it's going to take a lot of effort, I think.
Donna, those larvae,
what are
they
they-
taste good.
What
The
are they
ones in your
feeding on?
pantry?
My food in my pantry.
Those larvae of those butterfly moths, I think they taste good. Good
Okay.
cookie.
Tastes good. Okay.
We're talking Stuart here, right?
Yes.
Yes.
So hang on a second, everybody. Let me check in with Sean and see if he's all ready to go here.
Yes, I was just trying to move things around slightly here.
Okay.
There we go. That might be better.
And so you're familiar with the Zoom controls and you're good to go?
I think so. Yeah. Yeah. I did a little quick trial run, sharing my screen with my wife, and it seemed to work. So
Okay. Okay. Perfect. All right.
now that I'm doing it for real, it won't, right? But yeah.
Well, obviously, we're just yakking about LEPs right now, so join in if you like. Richard Ewell thought it was pretty funny that we were doing a whole presentation on cabbage whites.
I wish people would stop calling them cabbage whites. Small whites.
Small cabbage whites, okay?
Well, they're the English, you know, were long studying butterflies long before we did, and they called them small whites. Of course, they have a large white, which is in contrast, but the cabbage white is so indistinguished. You know, it's like,
Well, we could call them by their Latin name too, Jonathan.
no, we can't be doing that. That'll scramble our brains, I'm sure.
Or we could call them what a lot of gardeners call them, cabbage moths.
Yeah.
I'm always telling- no, they're
Yeah, a lot of-
butterflies.
Yeah, I like New Zealand. They call them the great white.
The great white, huh?
Yeah, because when they're introduced, they had a introduction. Actually, when my wife and I were on our honeymoon, they were battling them to try and not to let them get a foothold in their country. So they thought that if they marketed them to the public as the great white, you know, like a great white shark, then it might help
Oh.
increase efforts to catch them. So they actually paid people, I think it was $50 a pop, to go out and catch them, and they were successful. They caught hundreds, and they stopped the invasion.
So they essentially eradicated them from New Zealand?
Well, they hadn't been there. They were just introduced in like 2014 or something, 2015.
Oh my.
And so it was early on they caught it. They had, you know, a couple observations, or maybe an entomologist or someone collected them, knew that they had been introduced, and so they put out a campaign to raise public awareness and try to get, people to cite them and collect them early. And that's really the only way you're going to stop an introduction like that. Usually, once they get a foothold and spread, then it's almost impossible.
Those Kiwis are really serious about invasive species. They take it very, very seriously. They've gone on campaigns to kill all kind of critters that have gotten some blowback, but they've done it.
Yeah.
Well, what
Yeah,
I've heard from people who've been to New Zealand is that everything you see is a non-native species.
Really?
Well, the sheep and all the landscaping, most of the
right.
trees are non-native. Their forestry trees come from the West Coast, Douglas fir and Monterey pine, because they cut down all their own. And all the plants you see around any kind of human habitation, it's all non-native stuff. That's what I hear. I haven't been there. I wouldn't know what was native
in
there.
urban environments, that's probably true, like everywhere else in the world.
Well, yeah, like Seattle.
They don't have a lot of insect diversity there. It's just because it's too young, I think, of an island. So it's not like Australia. But yeah, I think the bird population is what they were most concerned, because a lot of the mammals that were more controversial in the eradication, like the rats and I forget what else.
Feral cats.
Feral cats and other things were decimating some of the native bird populations. But
well, they've got some of the best butterflies in the world. One of them is one of my favorite butterfly scientific names. It's Gonerilla, which I suppose could be pronounced gonorilla, but when I give presentations, I always pronounce it gonorrhea.
Of course you do.
Of course I do.
It does.
Well, I actually saw monarch larvae when I was in New Zealand, near Wellington, in a botanical garden. And it's the only time I've ever seen them in the wild. So that was kind of cool.
Another non-native species.
They've got great butterflies. The ones that are in New Zealand are amazing. They've got some coppers that are just knockouts. I mean, I've only seen, specimens in pictures, but
So Richard, I was telling them that you were laughing because we were having a presentation all about the cabbage white.
the small cabbage white, or the small white.
Or the small white, yes, as Jonathan is calling it.
Or the white rat, as I've heard it called among some Lepidopterus. But it's an interesting butterfly.
That's where I started objecting, because I live in the Fremont neighborhood of Seattle. That's why they were asking me if I saw cabbage whites lately. Because if it weren't for the cabbage white, I would rarely see a butterfly. I see tiger swallowtails in June, and in August sometimes I see the
Woodland skipper.
yeah, and then occasionally I see something else and I get really excited.
Yeah, that is one of the silver linings.
Well, I got excited when I saw that Sean was giving a presentation tonight, because he wrote a paper called The Global Invasion History of the Agricultural Pest Butterfly, cabbage white, revealed with genomics and citizen science by PNAS. I thought that was a great paper, Sean.
Oh, thank you.
Yeah, it is a great paper. That's how I found Dr. Ryan. That marvelous paper that he wrote with his co-authors. So that's some of what he's going to talk about tonight. So with that, why don't we go ahead and get started. I'm Paulette Murphy, the program director for the Seattle chapter of the Washington Butterfly Association. And for any of you who are not familiar with it, I want to, let me see, I need to share my screen first. I'm not, can you see my screen?
No.
Okay, well,
I can see
will
you.
try to figure that out. So the address for the webpage is wa.butterflyasof.org. And on our webpage, we have information about our meetings, both in Seattle and in Spokane, and about field trips whenever we're able to start having field trips again. And all of these are free, but if you would like to join us as a member or to donate, there's a tab that you can use to give us a contribution, and that can be done online. So tonight's presentation from the Seattle meeting will be by Dr. Sean Ryan, and the topic is how genetics and citizen science can help us understand insect ecology and evolution in a changing world. Dr. Ryan received his PhD from the University of Notre Dame and then went to a position as research geneticist at the USDA Department of Agriculture in Gainesville, Florida, in the fire ant division of all things. And from there, he went to another USDA position, a postdoctoral fellowship at University of Tennessee. And just last year went to join the scientific consulting company Exponent in Menlo Park, California. And tonight he is joining us by Zoom from California. And we want to thank you for joining us tonight, Dr. Ryan. Is it okay if I start recording your presentation?
Yeah, that's fine.
Okay, wonderful. Thank you. It's all yours then.
Well, thank you for inviting me. I'm honored to be here and have a chance to share some of the research that I was doing, I think now for the last 7 years. So some of the things that I'm going to talk about tonight started 7 years ago now. And so it's just fun to get to talk about, some of these things because you spend so much time working on them. And these days, since I'm in scientific consulting, I don't get to spend quite as much time working on this. It started as a side project and it's kind of gone back to a side project, some of the things I'll talk about tonight. So it's just great. It's also great to take a break from the election right now. So if you want to turn off your phones and just relax for an hour, if that's possible, and just enjoy talking about butterfly science, all kinds of things, other than the election. So let me start by sharing my screen. That'd probably be helpful here. Here we go. All right, can everyone see? Cover slide here. Wonderful. Talk a little bit about genetics, citizen science, and how we can use those tools to better understand insect ecology and evolution in a changing world, and have this fun little summary here. And I almost wasn't going to talk about everything in here, but I think tonight I will, if I can remember to touch on all of these. So just a little teaser of what I'll be talking about. So this talk features stories of invasion, conspiring with Russians, the potential breakdown of borders, and the recruitment of a worldwide network of citizens united to monitor one of Hitler's allies. So let's dig into what I'm talking about here. So I think when I began my career as a scientist or getting into grad school, I became really interested in how human activities were affecting our ecosystems and biodiversity more broadly. And I think it's pretty overwhelmingly clear that, through changes in our human activities, changing the climate, spreading invasive species, changes in land use, or dramatically altering our ecosystems. And this also includes insects, which we care about here in this group. Some have suggested that we're entering the insect apocalypse. There's debate, I think, about to what degree insects are in decline, but I think it's pretty clear, and there's pretty much agreement, I think, that many insect species abundance and diversity is declining, and it's just to what degree. And so there's just a lot of reasons to care about these, what we might call anthropogenic disturbances, and what impacts they're having on insect populations. And that's kind of what was driving my interest when I entered grad school. So here's my research in a nutshell, or really a Venn diagram. And so this is, when I have all of these circles overlapping, that's what I would consider the most interesting, most fun work that I've been doing. And that's, some component of entomology, ecology, and evolution, anthropogenic disturbances, and now more recently incorporating citizen science. And so
John, I'd like to interrupt for just a moment. We're not seeing the advancing slides, and it would be helpful if we could see it in the slideshow view as well.
It's not working?
We're not seeing the advancing slides. We're still seeing your title slide, and we see your your whole screen with the presentation thumbnails on the left and a bunch of stuff on the top. And I don't know if it's possible to present it in a slideshow view so that each of the slides is bigger.
Yeah, let's see. That's what I did, and I guess it just didn't translate. Let's see.
Yeah, you shared your PowerPoint, but then you didn't share it once it went to slideshow.
Oh, I see. I have to go. Yeah, I have to change it because the display settings automatically defaults. Let's see. Here we go. This should work.
Yeah. For a second.
Duplicate slide. There we go. Right.
Yeah, that works. Okay. Now we have a slide.
The Venn diagram.
We have your Venn diagram. We didn't see your second slide, but we see your third slide.
So now you guys are following along.
I think we might be. There we go. The Anthropocene. Yep.
Oh, sorry about that.
That's all right.
That'll make it more clear. Yeah. So here's just that slide that you missed, but yeah. So when all of these areas overlap, that's kind of where I'm happiest in terms of the research that I was doing. And so what I'll talk to you about today is kind of some of the work that's right here at the center of this Venn diagram. So when I started my PhD, climate change was a hot topic. So here you can see, changes in climate over 1960 to, projections into 2100. And, even by 2010, I don't know if my mouse works here, if you can see, but there was already quite a bit of warming that had occurred, and that's only increased since then. And so I was very interested what the impacts of climate changes were on insect populations in terms of ecology and evolution. And what we knew about climate change was that many species would be changing their distribution. So here's an excellent illustration of that. Obviously, it's with birds, because with birds we have some of the best data historically, and citizen science. So most of this is citizen science generated data. And you can see the rough grouse and projections of changes in its distribution as the climate warms. So that's, generally shifting further north. But there's also changes in the timing or the phenology of organisms. So you're probably aware that as the climate warms, many plants and animals might be doing things earlier in spring and then doing them later into fall. And so the timing of their events is also changing. But then the last one that I was most interested in was evolution. So are organisms evolving in response to changes in climate? And so at the time, 2013, so I was about 3 years into my PhD, and looking into the literature, there was a review that was done that basically showed that, perspectives on, the evolutionary responses to climate change had been growing, exponentially, but evidence had been lacking, or at least lagging behind. And the primary reason for this is, or at least one of the major reasons for this, is that we just don't have the data sets that we need to answer this question. So you need to have collections of individuals at, preferably at the population level, where you can look at changes over time. So you'd have to have them collected over a period of time and either measure them in a way where we can assess evolution, or have kept those collections so we can dig back in and look at them using genetic techniques. And so I was really excited when I got to work on a project. And this is for those that, may be happy that there's another butterfly other than the small cabbage white that I'll be talking about at least briefly. And that was with the Eastern Canadian, or the Canadian tiger swallowtail and the Eastern tiger swallowtail. And what was great and really interesting about the system and why I started working on it was because there was an entomologist professor at MSU who had been collecting these butterflies from across the eastern United States, and primarily in Wisconsin and Michigan, for his entire career. So from the 1980s, he had collections up into the early 2000s, and so we could actually use that as a window into the past of what was happening and compare that to what we were seeing in the contemporary samples. And so what I did for that work was, here you can see this yellow distribution. This is some modeling that we did to look at the northern range, or really the range of the Eastern tiger swallowtail, because it's driven mostly by climate. I don't have time to get too much into it, but I can answer questions later if you're interested. But really what you're seeing here is that the top part of the yellow here is the northern range boundary of Eastern tiger swallowtail. And then right where that ends, it abuts the Canadian tiger swallowtail, and they form a hybrid zone. So they're closely related and they can exchange genetic material. And so this was basically, we did a bunch of rearing, thousands of tiger swallowtails. Anyone that's raised the tiger swallowtails know that's a huge feat, because these things eat black cherry leaves. So it's like, deforesting a small area just to feed these guys. And what we did was we used that to parameterize models, where we then simulated how this distribution, or this hybrid zone, was going to change as we basically turned up the dial and warmed the climate across this region. And like I said, what was really cool was that we were then able to dig into Mark Schreiber's collection and actually generate some genetic markers, looked at what they look like genetically in the past, and then compare that to the contemporary time period in 2010. And what we were able to see is that basically what we were simulating, what we were seeing, the change in the movement of this hybrid zone, was confirmed with genetic data. And that was really cool. It's also in this PNAS paper. Exciting to get tiger swallowtails on the cover of that journal. So shout out to them. But what I also realized in doing this work was that, so Mark Schreiber had this collection. It was great. It was one of the best, that was available, but it was still not great. When I get to dig into the data, I'm like, we had some in Michigan, but through freezer failures and things, we lost them, but there wasn't much to begin with. Wisconsin, we had 1980, but we didn't have anything in the decades in between then and when I was, the time period, contemporary time period. And we didn't have the northeastern and region of the United States collected in a way that would be useful. So it's kind of like, I wish we had better sampling. And so the reason why I talk about this project, which was my first project in my PhD, is because it was really what was the impetus for what became the project that I'll be talking about at length today. And that is, what we needed was, long-term collections at the population level, preferably over a species, range, or a large portion of its range. If we were really going to, look at how species are responding to climate change, and here we have this natural experiment, the climate is changing. It's not whether it's going to, but to what degree it will. And so, we need to be collecting some of these species or organisms, or at least cataloging genetic information, so that we can actually look at, the evolutionary responses in real time. And so at first I thought, well, I'll create a citizen science project. Citizen science, I was getting involved in citizen science, involved in a working group with some of the leaders in the field. It was a really awesome experience. And with that, I was excited to tap into the power of the public to help with this. But then I realized catching tiger swallowtails is pretty tough. And because I was overly ambitious, I thought, well, they're really only in the eastern United States and they're not all over the world. We need some species that has a global distribution. And so what I ended up coming to was the small cabbage white butterfly, and for a number of other reasons. So the small cabbage white butterfly is found everywhere. So the eastern tiger and Canadian tiger are pretty abundant, but not to the level of the small cabbage white. I would argue that they are the most, or one of the most abundant butterflies on the planet. They had invaded, as I'll talk about in a minute, many parts of the world, but it really wasn't well known how that had happened, or at least the routes. And they're easy to catch compared to a tiger swallowtail. And so I thought, well, this is a perfect species to focus on. And so many people think these are just a vanilla butterfly, but they're actually a devastating agricultural pest to anyone that's had a garden, tried to grow any of the crops that are their host plants. And so they love crucifers, so they like cabbage and broccoli and kale. And so you can see, it's not a great picture, but you can kind of see the level of damage. And I've seen some really funny YouTube clips of people losing their minds about the small cabbage white butterfly and what it's doing to their garden. One interesting story that I found, and this is the case in working on this project, I found out a lot of these historical kind of gold nuggets of things that were going on that mentioned the small cabbage white butterfly. And one of them was this Dig for Victory campaign that was going on in World War II, where in England they were asking, the public to basically convert their lawns to gardens during this time of extreme rationing of food to help in terms of growing more food. And apparently the small cabbage white butterfly was devastating a lot of the crops, the cabbage that they were growing. And they didn't have BT back then. They didn't have very good methods. And I saw one of the methods was like taking ash water and putting it on the plants and things. And so they didn't really have a good way to stop them. And so one newspaper put out an article that said that that these cabbage white butterflies were one of Hitler's allies, and it was the duty of every student to go out and collect as many as they could. And so in this one a town, and I don't recall, but they had a fair, and all the students brought in their collections, and the winner had collected, I believe it was 10,000 small cabbage white butterflies. Second place was like 2,700 or something. And so that just gives you an idea of how many there were, but also how devastating they can be as a pest if unchecked. And so here's a distribution, the current distribution of the small cabbage white butterfly. This is not, like, the red isn't exactly where they are. It's if the country is filled in red, then that means they're found in that country. And there may be countries that they're found in, they just don't have confirmed observations or collections. And what we knew is, getting into this project, is that they likely originated in Eurasia, probably Europe. And then we knew that they were introduced in the mid 1800s. We had really good records of that, which I'll get into a little bit later. And then, about the mid or the 1930s, they popped up in New Zealand, and then made their way to Australia. And just to give you an idea of why it's no surprise that they made their way across the world, this is modern day shipping routes, but they were some degree of this in the past, and that is we just move people and products around quite a bit, and as we do that, some organisms hitch a ride. And so it's no surprise that the small cabbage white butterfly was one such species. Now you layer on top the, flights around the world, and we've been moving species well beyond the distribution or the dispersal capability that they would have naturally. And so as a result, there are now a growing number of invasive species. Invasive, different from non-native in that these species are argued to have a negative impact on the ecosystems or the economy. And so some of these species proliferate and they have these negative impacts. Cabbage white being one. But so we have this interconnected world. We're moving things around. We're inadvertently moving species that some of them have a negative impact, and that's unfortunate. But what's great is that we do have this well-connected world through the internet where we can connect with each other at a speed and scale that has never been possible. And so this is a huge opportunity to connect with the broader public and engaging them in scientific efforts. So citizen science is one way to do that. And really, this is the way I see it, is this is just scientific research that involves anyone who's just not a professional scientist, at least in that discipline. And so that can include scientists. This is one definition. There are many. I think this is a common definition, commonly accepted among practitioners. Now, community science is another term that is coming up, but it's basically engaging the broader public in the scientific process. And so I think one of the quotes that I like best by Jennifer Shirk is that citizen science offers the power of science to everyone and the power of everyone to science. And that's really why I got interested in engaging the public, or at least one of the reasons. And so from there, I was excited about citizen science. We had a species that I thought would be good to create a project where we basically asked the public to help collect some cabbage white butterflies from all across the world. Started this project and slowly began to put together a team of other graduate students, some at the University of Notre Dame, some at University of Nevada, Reno, Colorado State, and then even other researchers, professors from across the US and the world that you'll see in a minute. But so we started off as grad students, just trying to get this project up, get a website, and get the word out. So we started with creating a Facebook page, creating a Twitter account, and then trying to reach out to citizen science groups, reaching out to local media, to national media, just get the word out about our project. And there's a lot that could be said here. It's a lot of work. That some of these efforts were more successful than others. And so, like for example, we did an Ask Me Anything on Reddit. I don't know if you're familiar with Reddit, but it's a community where you can, propose, a topic and then let people just ask you questions. It was great. We made the front page because it happened to be at Notre Dame's football was, a hot topic. And so that worked well for us. So we got thousands of people to come to the website, then come to our project, but it translated into almost no contributions to the project. Where we really got the most success was, does anyone guess? Guess I can't hear you, but it was entomological and lepidopterist societies. And I think that might be no surprise to all of you. And that was that, people were very willing to help with our project. And so that's where we started getting the most success at first. But then we started getting more success as well from the broader public. And that project's been ongoing. That was, I think we launched that in 2014 and still going today. So we have over 3,000 small cabbage white butterflies that have been collected, half from 34 countries. I think we're at 35 now countries in counting. And half of these were collected by researchers and half by citizen scientists. So here on the right are some of the other scientists, researchers that were interested in the project. Roger in particular, who had collections from many places we didn't. It actually was perfect. It fit where, you know, where we didn't have samples, he had samples, and vice versa. And so this is, at the time, this is, these, you see these circles, the larger the circle, the more samples from those regions. This is kind of the sampling that we had at the time of turning these samples into to answering questions. But before I get there, when I started this project, I actually didn't have any money. So it was either stupid or bold. Now that I'm talking about it to you, it means it was bold, right? Because we made it, but we didn't have any funds, and crowdfunding was also popular at the time. So we thought, well, we'll just see if the public would be interested in helping to fund this project. Luckily, we were able to get it off the ground. A lot of it was from family, the helping out there. Crowdfunding is also really tough. But that small amount of money that we were able to generate helped us get more funds from University of Notre Dame, from various centers and initiatives. And then ultimately, I was able to get a USDA NIFA fellowship while I was working at the USDA. And that allowed me to, as a fellow, work on this full time. And so with that and some funding, we were able to sequence about 600 of these butterflies from all across the world. And so I say DD RADseq, this is just jargon here, but really the way we can think about this is that we have the genome of the small cabbage white butterfly. We know now, and we kind of knew then, that it's about 450 million base pairs. So you can think 450 million ACGTs lined up. And not to get into the details, but it costs more money if you want to sequence more of that genome, not just a small part, but all of it. And if you want more certainty that what you're sequencing is correct, because there are errors, that costs more money. So one way to get around that is just to sequence a subset of the genome. So you could think of it as like pointillism, those pictures where you have just points, and you don't need everything filled in, and you can still make out the broader picture. And so that's kind of what we did, is we sequenced, you know, subsets of the genome, so 22,000 markers spread across the genome. And with that, we were able to answer a number of questions. So it's a representation, a reduced representation of the genome. And so one of the first things we did was we looked at the genetic structure. And so you might have seen this with human populations, with like 23andMe or Ancestry or other projects. And what we did was we figured out that the first thing we did was that there was at least 7 genetically distinct populations across the world. And so you can see them colored here. They largely correspond to different continents, but you can also see some other interesting things here, like Siberia is genetically distinct from the rest of Asia, and North Africa is distinct from Europe. And then in North America, here you have most of North America is this blue genetic population, but you have this pink one here in the Western. This is like Northern California. That's also similar to what we find in Hawaii, and you know what's going on there. And then in Europe, what was somewhat strange is that because we had decent sampling there, we don't see any genetic structure within Europe. So we were hoping that we would see that, you know, England might be different from France and different from Sweden, different parts of Europe. Just like, you know, if you looked at 23andMe, you have these different regions that are genetically distinct for human ancestry. But we didn't see that. And the question is, why is that? Why is it what we would call one panmictic population that seems to be all well mixed? Could be that there's just really good dispersals through there. So you have a lot of connectivity. So humans could be helping and that dispersal. But there's also some evidence that the small cabbage white butterfly is migratory in Europe and possibly out of Africa. And so that could be one way that the populations are mixing over time. As the populations from the south migrate north, they may be mixing genetically and keeping them from becoming genetically distinct. It's also the case if we sequence more of the genome, we might be able to get at more of this fine scale resolution structure. And so with that, I was going to let us take a break if we want to ask a few questions, and then we can dive into playing 25 in Pieris.
So I didn't mention at the beginning, but the way we typically run these meetings is to ask questions at these breaks by chat. just to minimize the people talking over each other at the end, if Sean is willing to continue, we can have a little bit more of a free-for-all. So if you do have questions, please enter them by chat and I'll read them out. I did have a question about how you selected the sampling sites for your DNA sequencing. You said you, I think, selected 7 sites. How did you manage that?
Yeah, so for like these populations here, the
Yes.
little circles. Yeah, so it was really, you know, we had to use with what we had. So we have a few samples now and some other spots that we didn't before, but this is what we really wanted to get is as many individuals from a few regions as possible, but spread out across, let's see, how would I say this? Basically, we wanted like 10 to 20 individuals from what we thought was like a distinct region. But what we had sometimes was just more of like a spread out, few samples over broad scales. That also makes it harder to infer genetic structure as well. So some places we have a better sampling of that and some we don't. But really it was partly a function of what we had to work with, because we were lucky that we were able to be this successful in having, you know, a global collection. But it was really just what do we have? And so we'll use that. And so that's kind of how you have sampling within a continent or within a country. But we also wanted to have as many distinct locations represented as possible. And then we had the you know, limited budget, so we can only sequence so many individuals as well.
But how did you select the particular sites that you focused on? Since you couldn't sequence the whole genome, did you have prior information to help?
Oh, like in the genome, what
Yes.
sites in the genome? Oh, so that is it's relatively random. So this double digest. So essentially, there's these regions of the genome that these enzymes, there's different enzymes that will cut when they see that pattern. And so this is a double digest, so there's 2 enzymes. But really what you need, all you really need to take away from this is that these enzymes will cut at that pattern, the DNA, and that's where we then sequence from where that cut occurred. And we know that these enzymes cut relatively randomly, but some cut more frequently and less frequently. And so you can pick the enzymes based on if you wanted to cut lots and lots of pieces, or maybe not as many. And so that's kind of, we don't really get to pick where in the genome, but because it's a relatively random sampling across the genome, then we hope to get a pretty good, and we do get a pretty good representation of the entire genome.
Okay, thank you. There's a question about a word that you use, panmetic, that we're not familiar with. Could you explain that to us?
Yeah, panmictic is just like that, basically that is one homogeneous or one well-mixed population. So in case of Europe, it's that we don't see like, so I don't show you here, but you can actually, this 7 different genetically distinct populations is like I said, a conservative estimate. They're likely more distinct in some places, but due to not having enough samples or enough of the genome sequenced, we aren't able to tell it quite as well. And we wanted something a little bit more conservative for this paper, for this analysis. But if you were to look at, I believe it was like Japan, Korea, and China are actually all genetically distinct. They're fairly distinct from each other. So that means that if you were to look at this, these are the assignments on the right. I don't know if you probably haven't, I should have explained. These are basically, each of these lines that goes across, it's a bar, is an individual. And each color is the ancestry that we would assign to that individual, the proportion of ancestry. So here in Africa, all of them get this cyan, but you see a little bit of cyan bleeding into, this is Spain here. These are from Spain. And so there's some of this, you know, African ancestry in the Spanish population. And the same is true when you hear in North America, you have this light pink over here that's kind of bleeding into what we see across the West. So there's some evidence of that population. And so So what you'd want to see is that if with further structure within the populations, that they would fall out and usually one group, like that all of the samples in Korea would look that have the same color, and that you wouldn't just get a complete mix of random colors. And so I don't remember what the original question was, but
Okay, there's another question about your tiger swallowtail work. Your data showed a shift in the phenology in one of our members, Dr. Robert Pyle, noted that in Southwest Washington over 40 years, he has documented about a month shift forward in the adult emergence time and a change in nectar in the major nectar source.
Of the eastern tiger swallowtail, you said?
With the western tiger swallowtails.
With a western tiger, yeah, for sure. I would think that many species. There's papers, some great papers out now, and Ohio has had some excellent data sets to look at changes in phenology over, I forget the time period, but decadal. You know, I want to say like back at least to the 1980s. I can't remember. And I think Maryland as well had some great data sets. And many species are shifting the phenology, their timing, based on climate. But there's also the, I think it was at the Annis swallowtail or the western. I think it was the Annis. That's also shifted its timing through volcanism, having more generations each year as well in California. And I can't remember. I want to say it was the Annis swallowtail, but maybe it was the western tiger.
Well, I think there are a lot of anecdotal accounts about changes in phenology, but I think one of the really powerful things about the work that you've done is to document it so clearly, both genetically and worldwide. It's really, really quite wonderful work. So I want to thank you for that.
Yeah, and if everyone sticks around to the end, hopefully I'll move fast enough, and you'll be interested. But I do get into some of the citizen science stuff that I've done to try to measure phenology, which I would think this group might be interested in, because I think there's a huge opportunity to engage the public in actually collecting this data, because we don't know it for a lot of species. So we have there's two ways to look at it. It's the observations. We see the tiger swallowtails coming out. Someone, ideally has some standardized transect where they're actually, collecting them at the same time each year, and we can see that it's shifting. But another way to get at it too is to do some modeling where we grow them in growth chambers, and we look at how long it takes them to grow under certain temperatures, and we can use that to predict, when they're going to be coming out. So we can actually, predict the future in the sense where we predict when these butterflies should be coming out if the climate is warming, or under a given year's climate, what should they be doing? And so I think that's really cool.
Well, if you don't have time to get to that today, then maybe you'll come back and join us another time. So please continue.
Okay. Yeah, so getting back, so we have these 7 genetically distinct populations. Like I said, this is a conservative estimate. Some of the countries in here are actually probably distinct, or in fact, we know some of them are. But this is like a coarse view of the genetic structure. And with that, the next thing we wanted to do is we wanted to reconstruct the invasion history. So what was this series of basically movement, migration of, population as a source for a new location introduction? Who was the source? And it's just like playing 23andMe. I call it 25 and Pieris. We're trying to figure out, we have populations in North America. Where do they come from? What's their ancestry? And so what we start with here is just an example where we would have that's Europe and Asia. We knew those were the two that branched off first from historical records. But this Siberia population that we now know is genetically distinct, where did it come from? Was it Europe or was it Asia? Some might think Asia because it's kind of closer, but there's a path here where it could have branched off and came, from this more northern route. from Europe. And so these are equally possible scenarios. And so what we can do is simulate what those populations would look like if we had this, let these populations evolve in a computer simulation laboratory. Under these two different scenarios, we could simulate what they would look like genetically. But we know what they look like genetically because we have the samples from these populations. And so we can actually compare what we observed to what we simulated. This is somewhat of an oversimplification of the analysis that we did, but it's essentially what we're doing is we're comparing what we simulate to be what they look like genetically to what we actually see when we look at the sequences of these populations, and whichever one has the most similarity between what we simulated and observed we get more confidence for that scenario. And so then we would once we know, let's say Siberia came from Asia, then we go, okay, well, what's the next one that we know? So here, fortunately, we know the timing of these events somewhat. And so we can know which one came next potentially. So here we'd have North America. And say, did it come from Europe or did it come from Asia? We could do Siberia if we thought that was plausible, and so on and so forth. So after we get a bifurcation event and we've found the one that's most likely, we then move to the next, and we move to the next, and we move to the next. And so this goes from, back in time to present day. And so we eventually will make this full tree where all the populations we find today will be present, and the most likely scenario of how those bifurcation events occurred. And I should say, Eric Humbert is the one that did a lot of the heavy lifting here. This is a approximate Bayesian computation, random forest. It's just a very savvy statistical way to compare these different scenarios, the likelihood of these scenarios. And so what we found was that about 20,000 years ago, keep in mind there are huge confidence intervals. I think it's like from 7,000 to 30,000. I can't remember, but their estimates as they go further back, their uncertainty increases. But we have a median estimate of about 20,000 years ago that we estimate that the population in Eurasia underwent an expansion. So the population grew rapidly. And it's probably not the case that it had anything to do with agriculture, because brassicaceous crops weren't really being cultivated till about 7,000 years ago. So it was probably that it was the last glacial maximum, which also occurred at that time. And so you have this ice sheet that's receding, and the population is probably expanding in response to that. And we know that's been the case for many other species, so it's a likely explanation for this population expansion that we estimated about 20,000 years ago. Then more recently, about 1,200 years ago, again, this is the kind of the best estimate or the median estimate. We then predict that what we see in Europe and Asia split off into two genetically distinct, lineages here. And what's interesting is that coincides with kind of the peak of the trade routes, the silk trade routes throughout Asia. So you can see here in this illustration, these are the sea and land trade routes. And what's really interesting is so what we have here is this Europe population, and the Asian population kind of matches where Europe and those trade routes begin. And what's really interesting is that in Crete, they actually look like, I can call them Asian, but we can just call them the yellow population here. It's really interesting that here this trade route, the silk trade route, actually made it to Crete back then. So possibly some further support for these trade routes, where these butterflies essentially traveled along the silk trade route across Asia. And so that's really interesting. It's also the case that during this time period, it's probably not that the cultivation of brassicaceous crops were responsible for them being more successful, because they had been cultivated for a few thousand years by then, but it was the diversification. So there's a rapid diversification going on. So people were selecting different varieties, more leafy varieties like bok choys and cabbages, and And so maybe that there was just more food source and more variety available for these populations. And that might have helped in their spread, the timing of when this occurred. Then Siberia is estimated to have branched off next. And so we actually estimate that it happened about 1700 CE here. And what's really interesting is that when I looked into what was going on during that time period, we find that there was the Siberian route that was developing. So we have trade from China up into modern day Russia that was just beginning to be established right before these we estimate these butterflies to come off from Asia up into modern day Russia, and even the town. So here up here is Yakutsk, and so that's just fun trivia. One of the coldest places on the planet, one of the coldest cities. Somehow they're living up there, but that town had just been developed prior to this time period. So it's really interesting that as those parts of the world were settled, the cabbage white followed. Small cabbage white. And then in early 1800s, we estimate that it was introduced into North Africa. Now, I should say these estimates, as we talked about a little bit earlier, there's some potential gene flow between Spain and Africa, and so it could be that this actually occurred a little bit earlier, so like 1700, 1600. But 1800 is our best estimate at the moment. And what's interesting is that that corresponds with French imperialism into North Africa at that time. So possibly through that route that they were introduced to Africa. I couldn't find any first observations out of Africa. So if anyone can find any documentation of when the small cabbage white butterfly was first observed, that would be great. I just couldn't find any in my search. And then we have the introduction to North America. So that we knew from historical records, which I'll get into in one second, that it was about the mid 1800s. And we're trying to test whether there was just a single introduction, so like one ship with a handful or a bunch of small cabbage white butterflies, or were there multiple ships coming across, each time bringing a few more individuals. And so, a long story short, we think it's the latter. We think that it was just a couple introductions, and part of that is because the genetic diversity is a little higher in North America than we would expect, given that what happens as populations are introduced. So you have the ship coming across, you have some cabbage whites in there. There's probably not that many that are unrelated from each other, right? How many could we possibly have that unrelated? A few dozen, few hundred probably at most that would survive and make it. And so it's probably the case that there were a couple ships that made it over, because each time the small group of individuals is introduced, you have what's called a genetic bottleneck. So you can think of like if you had a bag of marbles of every color in a crayon box, dozens and dozens of colors, and you were to take just 3 out, you're going to lose a lot of that diversity, right? And so from there, they're only going to have those 3 colors. Some of them might even be the same color, right? It could have had 2 blues and a red or something. And so you've lost a lot of genetic diversity. It takes a while to build back up again. And so we see it slightly higher than we would expect. That's one form of evidence. But also what we have here is Samuel Scudder. Don't know if you know the late Samuel Scudder, but he was an entomologist, very interested in the introduction of the small cabbage white butterfly that had occurred just a few decades before him working on this project. He basically set out on what was a citizen science project, in which he wrote a letter. They used to do that. Now it's all email. In the mid 1800s, so shortly after the introduction, and he asked other, entomologists and scientists if they had observed this butterfly and when. And so he got back a few hundred responses, to over 200. And with that, he was able to reconstruct kind of the spread of the small cabbage white butterfly across North America. And I've included this because I just think that it was so fun. I put it together. So this is his map, and I've just kind of animated it a little bit. And And so here we have, he predicted that it was introduced in about 1860 in Quebec. And then, so just for reference, the Civil War is about to break out. And Charles Darwin just published Origin of the Species. So just a little bit of context here. The cabbage white butterfly basically started to expand along the Northeast. And here he has this population that kind of popped out of nowhere, possible introduction, separate introduction. Then these two fronts kind of merged together, and then some possible independent separate introductions in Georgia and Florida. And then those continue to grow and merge together. And eventually, the details at the end aren't as good, but they make their way to the West Coast, including over in Seattle and Washington, and up into Canada. And so they've kind of conquered much of North America by the early 1900s. And so what's cool is that we've kind of shown that to be the case, and we'll get back to a little bit about how they made that their way. Actually, now we'll get back to how they made their way at that last jump there. So how did they this pink population, this West Coast population, it's primarily in Northern California, come into existence? Where did it come from? And so one possibility, Arthur Shapiro is great at butterfly knowledge and history and so many things. And he had thought, well, one potential possibility was that the Spaniards had brought them over early on. And so we looked at this possibility of this early introduction, even predating the 1860s introduction into North America. But it didn't look like that was the case. What it actually looked like was that it happened that the introduction came from the East Coast relatively early on, about 20 years after they were first introduced into the East Coast or Quebec. And then from there, they kind of spread out and into this little bubble that they are in now, and at some point into Hawaii. And so I was looking, well, what was going on? As I told you, the Civil War. So part of the Civil War, thankfully, the rail lines had been more developed in the North, and that's partly why the North won. And so I was looking at rail line development over time, and here you can see railroads being developed in the year they were being developed, and you can see here the North, thankfully, has more developed. We won the Civil War, but then you can see this little path rail line coming out to the West Coast, and shortly after it does, that's when we predict the small cabbage white butterfly to have been introduced. So I think that's as close to a smoking gun as you can get, that they basically hitched a ride on a train. Small group of them made their way over, and then they started a population that grew before the rest of that front moving from the East Coast made its way. And so they were able to establish a population that basically made it so that it was not as easy for them to be kind of swamped out by the genetic population that was kind of expanding across from the East Coast. So that was really cool. So they came by plane. Train. So if we can find any evidence of them going by train, we could have planes, trains, and maybe automobiles here. So then the question is, how do they get Hawaii? So we estimate that it actually, because we know from historical records that it was that happened in about late 1890s. We know that it came from this West Coast population and probably San Francisco. So there were ships going from San Francisco to Hawaii. And so through those shipping routes, they were probably introduced. And then from there, I shouldn't have given it away, but many people thought that, where did the New Zealand population come from in Australia? The thought was, at least my thought, and I think many others, was that it came from the UK. And that's just because there was a lot more trade going on between them. And, New Zealand is former colony, as well as New Zealand. And so, those are probably the source, but that's not what we find. We find actually that the San Francisco, again, likely introduced them to New Zealand. And then from there, shortly after, New Zealand introduced them to Australia. So that was cool and interesting. And so I talked about before, so here we have this is jargon. Just think here of genetic diversity high, genetic diversity low. So you're going from low to high here. And here we have the populations. So Europe, we think Europe and Asia is kind of where things started off. And then from there, this is the chronology of introduction. And what you can see is that they basically become less genetically diverse as each introduction occurs, with the exception of the US, which is a lot of variability and a little higher than we might expect. And then so what this is just a genetic, like a small group of butterflies that expanded and grew, and then gave a small group of them gave rise to another population, and so on and so forth. So it's a bottleneck within a bottleneck with a bottleneck, which is kind of impressive that these butterflies, even though they keep losing successively genetic diversity, they're able to continue to, be introduced and expand and be extremely successful. And so I was just going to do a plug here for anyone that's interested in helping do some additional collecting. So the project is still ongoing. We get actually a lot of samples from right now Washington and Oregon are some of the most avid collections we've had this year. We have people now, so we have this a great sampling, but there are still some spots in terms of like spatial distribution where we don't have. So here we have, this is a better picture of what that pink cluster I was telling you about, just at San Francisco, but it kind of goes a little into Southern Oregon and even into Nevada. But we don't know, what it looks like in these regions. How far does this pink extend? Does it go up into like Northern Oregon almost, or down into LA? Where does it end? And is there a hybrid zone between these two populations? We do have some evidence that they are sharing some genetic material. Good night, sweetie. Little one's going to bed. And so it'd be really great to have some collections in this area, and we've been able to start to do that. The other thing is that what would be great is, because we'll get to it in a minute, that we're also looking at morphological variation that changes across the season, from spring to summer. And so getting people to collect a few throughout the year, and then we can look at how that morphology changes over time. And so some people have volunteered to help with that. And then the other big thing is, which I'll get to, we have a break coming up, but I'll get to in a minute, which is just basically that what I set out to do when I started this project was not just to reconstruct the invasion history. That was actually the short-term project to get to a long-term goal, which is I'd love to be able to, now we have this great sampling in the 2015, 16, 17, and every 5 to 10 years, do another pretty good sampling over similar locations. So we can actually look, compare the same populations over time. And we can look at, just like I showed you with those moving of the bird populations over time in response to changes in climate or predicted changes in climate, we could look at them, over time, the genetic changes as these populations are likely to evolve or involve not just to climate change, but other things like the introduction of other host plants that they might be using, like garlic mustard or other things like that. And so we can document those if we have actual samples. And so I think this is the perfect species to do it. Like I said, it's abundant. It's it's an agricultural pest. Farmers are trying to get rid of it, and it's not really going to be really hurt in most of its locations if we focus on this species. So I think of it as kind of an ambassador to learning about butterflies. So that's just a plug that anyone's interested in contributing. We're always looking for more help, or just even spreading the word about the project. And with that, I can think it's a great time to take questions if you have any.
One of our participants notes that in Gerald Durrell's book, he mentioned that when his family moved to Corfu, he took, in quotes, a jam jar full of caterpillars. So it's possible that some kid could have done the same thing, jumped on the railroad and started the whole thing.
Yeah, point back to an actual family. We'll have them to blame. That's what I always tease, I missed it in this talk. I always tease that maybe it was my Irish ancestors that brought them over to North America to begin with.
I also wanted to ask about South America. It seems remarkable that you don't have any dots in South America, and I'm wondering whether that's a sampling issue or what you think, if you have any thoughts about what that might be.
Yeah, and great job paying attention. I was supposed to mention that. So South America and Antarctica are the only continents where they're not found. And I've tried really hard. I've talked with Arthur Shapiro, who knows probably more than anyone about this species, and others as well. And there are no observations of them. There's a large white that made it to Chile, and I'm not sure if other parts of South America, but somehow the small cabbage white butterfly didn't. And I bet if we looked at the the trade routes and the direction of those trade routes and things and what was being traded, there might be some evidence of why there just wasn't that pressure, what we'd call propagular pressure. But I did find it strange that of all they just haven't made it there. And I don't know what the contemporary efforts are in keeping them out, because you'd think that they could still be introduced, but they just haven't.
Okay, there's a question about whether you need collections from Seattle or whether you're particularly looking to fill in geographical areas that you don't have information.
Both. So the thing is, we don't have samples from the same location every year, because some people will collect in, like, let's say Seattle one year, but then they don't participate the next year. And so we do encourage people to collect them, just a few, even if we've already collected them in a previous year, because that way we can look at long-term changes. So especially in what I'm going to try to send out is like an announcement, maybe this year, because we didn't get our collection started until like 2015-16 as like a 5-year anniversary mark. But so it's still important to collect in the same locations if we already have them there, just so that we can look at temporal changes over time. But they also, like I said, if one can collect them just a few times in the season, even if it's just a single individual, that will help for looking at morphological variation, because they change quite a bit morphologically from spring to summer. And we're trying to develop a model to basically predict what they'll look like, given where they live, so the latitude and the temperature that they're experiencing. And so that will help us get a better handle on that.
Well, I wouldn't be surprised if you'll have some very enthusiastic participants following your talk here. Our newsletter director suggested that if you send a blurb about the project or what information you want to disseminate, that she'll publish it in our newsletter.
Oh, wonderful.
And then there's another comment from Dr. Pyle, who says that the letter that you showed of Scudder's must have been one of the very first citizen science initiatives in North America, and wonders if you would agree with that.
Well, I think it's a citizen science effort, and I think that you can argue that. The first one, I'm not sure. With the Christmas bird count dates back pretty far, and so they've taken claim to one of the first. But also the case of Thomas, like some might consider what Thomas Jefferson did. So the whole reason why we are able to predict the climate now at all, or weather, really, is because Thomas Jefferson sent out thermometers to people all across North America and asked them just to record the, Tmin, Tmax, the minimum temperature, maximum temperature in each day, and then return that information back. And with it, they were able to kind of get an idea of how temperature changes in one region were then subsequently changing in other regions, and kind of put together a very basic weather prediction. Obviously, that's gotten a lot farther now, but some argue that's one of the first citizen science. But it's definitely one of the earliest with Scudder, and maybe one could argue with the caveat that it's the first entomological citizen science project in North America.
Okay.
Kind of has us beat there in terms of entomological citizen science. They had one that dates back, I think, a few thousand years ago. So, but.
Okay, I think there's going to be some lively discussion at the end, so I won't delay you any further.
Okay. Am I doing okay? The last part should go pretty quick. Do I have?
Whatever time you're willing to give us, Sean.
Oh, okay. Oh, great.
Really happy.
But this stuff I think is fun. So I was going to share some of the things we worked on. So when I was in Raleigh, I was a citizen science fellow, the first citizen science fellow, speaking of firsts. And I was also a USDA fellow as a dual appointment, University of Tennessee, Knoxville, and North Carolina State University in Raleigh. There was 2 REU research undergraduates that were working with me. And so we had this data set. We have all this genetic data, genomic data of these butterflies, 600 butterflies from all around the world. And so we knew a lot about the genetics of the butterfly. But in each of those samples, there's more than just butterfly DNA. And that's because when you take a butterfly and you grind it up, there's all the things that were on the butterfly in its gut, and that those things have DNA, and those things are extracted in the process, and we get sequences of those things too. Not as many, because we didn't try to get those things, but they're still there, just kind of accidentally. So often those are considered contamination, maybe, right? You just things you don't care about, you don't worry about. And someone else also call them eDNA or environmental DNA, just DNA that they're picking up. But it's in these samples potentially. And so we were interested in looking at, well, what do we see if we look in there? So we find all these other organisms that can potentially be associated with the cabbage white butterfly. And so this work is still in progress. But basically what we did is we take all of the sequences that we had and we map them. We map them, basically we align them to the genome of the cabbage white butterfly. So we say, okay, we're pretty certain this goes to the cabbage white butterfly. Those were used in the study that I just talked about. Let's throw those away. What we're interested in is we take what's remaining and we look at this, tree of life, so to speak. This is basically the genetic data that we have for all the species. Keep in mind that we don't have a lot of genetic data for some species. So this is like, you can think of the tree of life, all these leaves are genetic sequences for different species. Each leaf is a different species. If it were to mimic, all the different branches on the tree of life, a lot of them are bare, sadly, or spotty, and some are more overrepresented than others, but it's the best that we have. And so we can take those sequences, we can say, okay, what is this best match? And we can get a decent idea of what species that sequence might belong to. And so what we found at first is that, when we look at we did that, we pull out the plants. Okay, what plants are there? And what we find is kind of a who's who of agricultural crops. And this is pretty cool because this is kind of what you'd expect. It's an agricultural pest. So these things are in, agricultural landscapes. This is where they like to be, at least for their host plants. And so it's not surprising that they're picking up DNA of other crop species, if that's what's going on, or possibly, so it could be DNA that's on them from plant tissue or pollen, but it could also be ingested through nectar. There's pollen sometimes in the nectar of the flowers. So, but it's not surprising. They tend to be associated with crop plants, so that we're finding a lot of crop plant DNA associated with them was kind of cool. What was intriguing was that when we looked at the bacteria and fungi, what we find is that many of them are plant pathogens or parasites. And so these are things that, cause a lot of harm to a lot of these agricultural pests. So these are often well known for their impacts on agricultural pests. So some of these are probably real. So keep in mind there's some false positives in here for various reasons. But I mean, if even a grain of this is true, it suggests that these butterflies might be spreading around plant parasites and pathogens if there's enough of that material to move it around, which was really interesting. What was kind of crazy and concerning was that when we look at the metazoans, or you can think of these as like the other animals, what we find is that we find some strange things, like Asian carp. So these are the Latin names here, but you got Asian carp, you have like a Ragalides fly, you have as a zebrafish, chimpanzee, mouse. What is that? As like a Chrysomya? I can't remember the common name. But these are species that you wouldn't necessarily expect to find DNA, especially Asian carp or zebrafish. So unfortunately, you're muted, so you can't guess. But where did these come from? these are all species, like all of these, or at least majority of these, are species that were heavily studied at the University of Notre Dame. So this is likely contamination. So that through the process of extracting DNA from these butterflies, we're picking up Asian carp. So there's a lab working heavily on Asian carp nearby, and so we're picking up some of that signal. Now keep in mind, when you look at these, this is the number of individuals that had number of butterflies that had that Asian carp DNA is quite a lot, right? 20%. But when you look at the number of molecules of DNA, they're very small. So it'd be easy to weed these out and say, oh, this is just noise, right? It's just 3 molecules out of, we're talking hundreds of, actually millions of molecules. So it's not something that's a substantial numbers, but it's kind of interesting. And what I was thinking is that this isn't unique to Notre Dame. This isn't that we just do bad, sloppy science and we're picking up DNA of other organisms. This is in most data sets, probably every data set, genetic data set. And then it's just that you get a little bit of DNA that's floating around everywhere. And so it's going to make its way into your sample. And I would propose that if you took a swab, a Q-tip, and you swabbed the hallways of a research lab and you sequenced it, and you knew what organisms they studied, you might be able to figure out where that swab came from. And so it's kind of like a forensics of where did this batch of DNA come from, based on all the little other molecules that we're finding in there. So that was interesting, but it raises the concern that one needs to be very careful in interpreting some of these results and making sure that they're not just a contamination. And then we were looking at Wolbachia. So Wolbachia is this endosymbiont. It infects, what is it, 25 to 75% of our arthropods. We don't really know how many. There's actually a really cool citizen science project, maybe as cool or cooler in some ways than the Pieris Project now, is that they're asking people to send in insects, any insects. I think they've been working with schools, and they'll extract DNA from them, and then they'll look for, they use a really well-developed genetic marker that they know is for Wolbachia. And if they if you find it in there, then you send it off to them and they'll sequence it. And so they're trying to get a handle on all the different insects and at what frequency they have this endosymbiont, because it's one of the most commonly infected in insects, and maybe helping some insects, but it also does some really weird stuff. Like if one is infected with Wolbachia, one individual, like a male or a female, one has it and the other one doesn't, it messes with their reproduction. So it either kills males or it feminizes males. And so it does this really interesting effects on the reproduction. And it's actually being used here in California. Actually, there's a company that is using it to try to control mosquitoes. So by infecting mosquitoes with Wolbachia and then releasing those out into the wild, they're basically, the males will mate with the females and then they won't produce any offspring. And so it's a way of kind of bringing down the mosquito populations without having to do like spraying pesticides or anything like that. They can just basically give these mosquitoes Wolbachia and then kind of flood the females. They do that with sterilization too. They bombard insects with radiation. This is just another way to do that. And so what we were looking at the individuals in our samples have Wolbachia, and we found some interesting results that it seemed like we find a few individuals in each of these populations that have Wolbachia. And so we need to follow that up and confirm. But if it's true, it's, what is it doing in these butterflies? Does it have some effect on the reproduction? What is the invasion history of that? Where did it come from? Does it match what we see from the spread of the butterfly itself, or is it different? And so that's one of those things to follow up, but really interesting that we see evidence of it in the small cabbage white butterfly. And then as I was saying earlier, that these are the things that we've done since as kind of like side projects or work in progress after the invasion history. And like I was saying, I think there's a great opportunity now. We have collections from all across the world and each of these continents, and we can essentially look at how these populations are evolving over time. But we can also look at, we don't even have to wait into the future. We can look at how these populations have kind of evolved as they spread across each of these different continents. So as they were introduced into North America, they started off in Quebec, but then they occupy such different climates, right? From, Alberta to Mexico. And so they've likely evolved, almost certainly have evolved adaptations to, be better fit in particular regions. And so we're working on that now to try to look at some of how these populations may be evolving or have evolved as they spread out across the different continents. And then looking at the phenotype, as I was saying earlier. So here you have the butterfly wings. So we try to make the most out of every butterfly. They've sacrificed a lot for our project. And so we basically photograph each butterfly, dorsal, ventral. So this is, let's get Andrew on here. Andrew Stower, who's actually been photographing all the samples that we have. And he's done a lot of his own photographing of collections he does in Indiana, where he's kind of like hyper focused on doing a really good job in one location. And so one cool thing that we're working on was creating the Zooniverse. I don't know if you're familiar with Zooniverse, but it's an online citizen science platform where people can basically contribute to digital citizen science projects. Often it's image based. So you're looking at images for like penguins, count penguins, or look at camera traps in the Serengeti and things. And so we were going to have people help measure some of these traits, like how large the right forewing is, the size of these dots. And it's because these things change throughout the season, and then they may be changing or different between or among the different populations. And so so that's a citizen science project we're going to hopefully launch and get the public to help measure these morphological traits. And then Anne Espacet, who was one of the original grad students on this project, who helped immensely in getting this kind of off the ground, was looking at the white pigmentation. So Andrew's interested in the melanin, the black pigmentation, because that changes quite a bit across the season. So they become kind of lighter. So they're darker in spring and they get lighter in summer. And Anne was interested in the white pigment, pterin, which is this the white pigment in the wings. And they had done some research and knew that the amount of nitrogen that they feed on actually increases the amount of pterin in the wings. So they're brighter. And this is important. So here you have a male, and basically they're using that as a way to signal that they're good. They had some really good amount of nitrogen, which they pass off in their spermatophore, which is good for the young. So the females are selective and they choose the brighter females. There's a great article in the Atlantic by Ed Young. I believe it's called The Extraordinary Sex Life of Butterflies, something like that. And it's about the small cabbage white butterfly. It is a wonderful read. And it talks a little bit about this and Nathan Morehouse's work specifically, which is related to this sexual signaling. So she was looking at if she could find a signal. We have these collections from all across the world, and see if in agricultural environments where we tend to have more nitrogen, we're adding more nitrogen, if she sees a signal from that. And so that work is nearly done, and hopefully we'll have something to share soon. But so she's measuring that. So we're trying to get a lot out of all of these collections. And then to change gears just really quick, the last thing is I'm interested in phenology. So we talked a little bit about phenology early on, but this project hasn't really done much phenology. Tiger swallowtail had a lot of phenology related to it that I didn't get to get into, but I'm very interested in phenology and how climate drives phenological changes. So the timing, again, phenology is just the timing of when organisms are doing things. And so I'm interested in citizen science. I think there's huge opportunity to get the public to help with this. So I did all this rearing of these tiger swallowtails, thousands of them. But there's so many species out there, and they differ in different locations. So we have people, the tiger swallowtails, the eastern tiger swallowtails in Florida are going to be different than the ones in Illinois, right? So they might actually grow a little faster. So we would predict, differences in their phenology, depending on like the climate that they're receiving or the temperature that year. And so, we really want to get at all that variation within a species, among species. And we want to connect the public with butterflies and with science and climate change and phenology. And so there's a huge opportunity to do that. And so I started a project where we basically ask the public to help collect or help to raise caterpillars. So I went out and collected an American lady, and we had cabbage whites, and basically gave them cups. So I worked with some local schools, and just basically said, okay, here we have, raise them from each stage, and monitor when they go from egg to caterpillar, caterpillar to pupa, pupa to butterfly or chrysalis, and just record that, and then the really only other thing we need is to have a data monitor in there, like a temperature logger. And so we log the temperature. They don't have to worry about that. They just record those observations, and with that we can calculate what's called degree days. I won't get into it, but it's basically just a measure of if we know this, they receive this much temperature, this is how much they grew, and how much of their development they completed. And so, like I was saying, I was actually working with a local high school student on this, and so we work with local schools. We worked with the Museum of Life and Sciences in Durham. I don't know if you've been there in North Carolina. It's amazing. We really miss it for our daughter. It's a cool place to be. But they have a huge tropical butterfly greenhouse, and they're really interested in outreach. And so they help fund this as well, just to get this going, where we had people were willing to pay to participate in this project. So I know there's huge demand. People are interested in wanting to raise a caterpillar and learn a little bit about the butterfly and contribute to science. And so it was a huge success in that way. One of the cool things we did was, I think we talked about this a little bit before my talk, which was the Cabbage White Butterfly. Someone had mentioned that in Seattle, it's like one of the few things you get to see, right? And so one of maybe critique's too strong a word, but critique of these tropical butterfly gardens or greenhouses is that when people leave, they're never going to see a blue morpho in the wild, and so they're not going to be able to connect, when they leave. And so I proposed we would let people release the butterflies that they had reared, so the small cabbage white butterfly, and it would have been the American lady, although I moved to California, as you heard. But just to have some, local species included in there, and they could learn a little bit about species that they're going to see when they leave the museum. And so we had them release them. We also didn't want to release cabbage white butterflies into the wild necessarily. And so one of the things we looked at was I reared some and measured them, recorded their developmental stages with the temperature loggers to compare it, because I'm supposed to be the expert. And so compare it, and what do you know? These were second graders. Second graders do pretty much as good a job as I do. So that's why I'm no longer in science. No. Yeah, so they do a great job. They get pretty good estimates, not significantly different than what I was able to do. So with that, for the small cabbage white butterfly, at least we could estimate these growing degree days. And with that, we knew how many, like if we had the temperature, we could predict how much development they would accumulate. And with that, we could predict certain milestones. If the temperature kept in, if we had the temperature profile for every day, we could predict these milestones. Like when are they going to basically become a butterfly? And so what I'm about to show you is if we take the climate for North America, I can't remember if this was the average from like the last 10 years. Apologize, I can't remember. We did some averaging of the climate. It's not that important. This was just kind of a proof of concept and just exploratory. But basically take that information that we know in terms of development and temperature, and then feed it, average temperature in North America. And what you're about to see is going to look like a migration. So the little white dots you're about to see are we're predicting a small cabbage white butterfly to emerge, and then the dot will glow, turn white, and then it'll disappear. And every time the model predicts a cabbage white butterfly at that day of the year, it'll turn white. So it's going to look like a migration, but it's really more like, think of a wave, like, one little spot. It's like, hey, cabbage white butterfly here, and then it's gonna go to the next one. And so you'll see here. There we go. So this is probably a little early. Like we're predicting a little earlier than it really is, and that's just because it's hard to get a handle on the date of first emergence. But what's interesting is you see that you have this like band. So now Florida's onto its 4th generation here, as that just up in, Canada, they're starting to see cabbage whites for the first time. And so you can see this like successive generations moving across North America, and that's just because that North America is warming in this fashion, right? It's going to warm from the southern states first as it gets warmer and warmer further north, and so that's why you see what looks like kind of this wave, this kind of delayed response, and then you have, this is cabbage white butterflies all across North America at the same time, being present later in the year, and then they start to fade out. What's interesting too in the Sierras, it becomes much more heterogeneous. It's just kind of all over the place, and that's probably because of the climate in those regions is just a little bit more variable. But just it was really cool, and so this is what I was hoping to do is work with the public if we can collect data like this for different species, and even fine tune it for different locations, right? So our estimates based on butterflies collected in North Carolina and actually a few like Indiana, but really we might want to fine tune it so that we predict slightly different timing for Washington or other places because they're adapted just a little bit different to the climate there. And so we can then start to build this like really cool predictive model of, okay, this year we predict that this is when the butterflies are going to come out. And so the National Phenology Network, I don't know if you're familiar with it, but it's a great citizen science project, and they do that. They have now a forecasting tool that they've developed for some species. They're really interested in the timing of when species are going to come out and trying to predict that each year. And so I was started to develop a project that I haven't gotten too far with, but it was basically called a disco bug, but basically trying to have something catchy, but just basically, just like with the Pieris Project, have a portal where people can come get involved, learn about different species and how they can get involved in collecting data that is useful. So I called it the temperature connection. So basically getting the data logger. And I got this really down to really cheap where we could, I could do a setup for like less than $1, I think. And then the data logger is what's, expensive, but we could probably, hack some of those and make some if we wanted to make them cheaper. But people can get involved with collecting this developmental data as it relates to temperature. They could do the sensitive insects. So one of the things that I thought would be really cool is that, as all of you probably know, but there's this critical stage in insect development where insects will become sensitive to the environment and decide whether they're going to go on for another generation or whether they're going to diapause and basically hibernate for the winter, essentially. And so we don't know that stage for many insects. But we could determine that by doing some fun little experiments where people put them in the dark for so many hours at different stages and see at which stage, when they did that, did they diapause versus come out again. And then what I call the herbivore's dilemma, where many species eat different host plants. But depending on what they eat will influence the developmental rates. So we give them, here's like the eastern tiger swallowtail, giving them ash versus cherry or something. We can actually expect them to have different phenologies depending on what they eat. So just different ways to get information. It's kind of like natural history information on different butterfly species that could then be used to learn more about them and make predictions of how they're going to change in response to changes in climate. Just thought it was a fun project, and it seems like a project that people were really receptive to, but haven't gotten too much farther than that since I've left North Carolina. And then so just my last slide is just a thank you to so many collaborators that have worked on these projects, and then the citizen scientists that too many to name that have contributed to this project. And so just a thank you to all of them, and then the funding sources for that work. And with that, I can take any questions.
Thank you so much, Sean. There are a couple of specific questions about this portion of your presentation. One is about Wolbachia and exactly what it is. Is it a bacterium or?
That's correct. Yep.
Okay. We just weren't sure about that.
Sorry.
And another question about whether you've looked at if you've interrogated the wings of Pieris rapae using ultraviolet light.
So how are they looking at that?
Yeah, there was an image there that we.
Yeah, I'm trying to remember if that image is taken through ultraviolet light. I should know that, but they are measuring that it is. Yeah, they are looking at the wavelength of what's reflected. I don't know if they're using a black light on them. I mean, I guess they use a multi-spectrum light.
Yeah, the UV is very low reflectivity. If you look down at 200 nanometers or something, I'm guessing.
Yeah, I can't remember the experimental setup for this, but they do look at the reflectance at different wavelengths.
Okay, well, Anne's work generated a lot of interest on the chat, so we may follow up with that because that sounds very, very interesting to look at the pigmentation. So this ends the formal presentation, Sean. So thank you so much. We're so grateful to you. And there are a couple people who haven't asked questions on chat, but one in particular, Jonathan Pelham, is the curator I mentioned at the Burke Museum. And Jonathan, if you'd like to unmute, then maybe you can ask whatever questions, and then everybody's free to unmute, and we can just have a general question and answer session or BS session or whatever folks want. So are you? Good.
I'm going. Hey, John, this is John Pelham. I really appreciate all that you're doing. I wanted to let you know that we have a very nice citizen science community in Washington, working on compiling records of butterflies. We actually are able to demonstrate those phenological waves that you just showed us, which was a cool graphic. And we can do that on a local level. We actually show the movement of butterflies from one part of the state to another over the course of either a season or as a product of migration. And as we compile these records and get them together, I'm going to ship them off to you. It'll be phenological data. We might be able to corral some specimens along the way. The only other thing I had to say was our small whites in this state, they are definitely darker on the ventral surface in the spring and paler in the summer, but on the dorsal surface, they're almost completely suppressed in the spring. You don't hardly see even a dot. We have another white species in the same genus, which is marginalis, and it is noted for being almost spotless. And actually, the earliest small whites will be confused for it because they too are nearly spotless. That's it, baby. Thanks.
Yeah, very cool. Yeah, so then that's one of the things. So there's tons of data too on the phenology. So those sounds like you're using observations to phenological events, so like butterfly sightings. Is that correct?
Well, we use sightings, I think, more and more frequently now because we have so many eyes. But the initial, the primary database is actually collection based.
Collections, okay, yeah, but so a physical specimen. Yeah, and I think that's the perfect complement. So the one that I showed wasn't even based on observations. It's just a prediction based on, we know something about their physiology, life history, and we're just predicting when they'll be out. And so perfect for that, for a validation, is if we have great data sets to actually say, okay, well, the model predicts that the butterfly should be out around here. Is that what people are saying? And so you can kind of use those, combine those to really basically inform or validate these model projections to have really robust predictions. So that's great. So yeah, combining those two is wonderful.
It'll be in your pocket pretty soon.
Awesome, thank you.
So your work on the citizen science, is that something you're hoping to continue with despite all your other challenges?
Yeah, I still get to do a fair amount of research. I was teasing, I said I wasn't a scientist anymore. I'd still do a lot of science. I just don't publish as much. And this work is just not quite in line with a lot of the things that we do on a day-to-day basis. But it's kind of a hobby. So now I guess I'm a citizen scientist driving a citizen scientist project. It's kind of my second baby, and so I can't let it go. And so I work on it a little bit, not as fast as I would have think if I was still in academia, but definitely keeping it going. And there's lots of collaborators that I've developed prior to the project and since the project began that are interested in leveraging it because it's just a great resource now. And we still get new people coming to the project. Since COVID, I think got more people like wanting to get out and do something. And so, yeah, I just think that it's great, and it has to keep going if we actually really want to do what I intended from the beginning, which is to look at long-term changes. And so I think it's a great system to do that. And so, yeah, hopefully at least every 5-10 years we can get a good sampling across the world.
Sean,
Yeah.
Bob Pyle, thank you so much. Wonderful talk. It's delightful to have this caliber of scientific and just natural history input in our many locations. So thank you. No, I've witnessed the full setback of Papilio rudilus, the western tiger, by a full month in 40 years in my location. I would expect an advance in phenology, not a setback. So that's strange. But I'm just about to analyze the data, and I may be in touch with you for your data. But I wanted to ask you, how do you organize your time between the citizen science, which, as I know, is very time consuming, and your own science, and B, I didn't hear your preliminary maybe, but if you've left academia, you were at Notre Dame, I take it.
PhD at Notre Dame, then I worked at the USDA, then I was a fellow through the USDA funding as a NIFA fellow, National Institute of.
Right. So what are you doing now?
Now I work at Exponent, so I'm a scientific consultant. So I publish, but I don't do, research that involves, laboratory collecting specimens. So this is all, for fun. But it's some. So I have a 4-year-old daughter, 4.5 now.
And where do you live?
I live in the Bay Area, so I work
Okay.
at a Menlo Park.
Okay.
That's going to be changing. I'll be moving down to the LA area come spring. Not sure when, given everything going on, but California based now. But the part of the reason, so this project is just like, it's just too cool. It keeps me sane to work on. It's just like why all of you are working on different projects, right? That you're not getting paid to. It's just like, it's something that it makes me happy. But it's also, I have a 4.5-year-old daughter. And so we have an opportunity to do research. We just got a dissecting scope, like a stereo microscope. And so we're going to go look at seeds and things. But part of it is I want to do citizen science. And the Pieris Project, we might have to wait a little while because it's like a lot of it's a little bit above our head at the moment. But the idea is to keep it going and keep working on it together. And so, yeah. I mean, one of the pictures in my thing was my mom. So my mom had collected some specimens, and yeah, it's a lot of work. So if your question is how do I do it, slowly. It just moves a lot slower than it used to, but
Well,
it still moves.
Look, when you get down to Southern Cal, let me put a word in your ear. It's not going to be plastics. It's going to be tailed copper, one of the coolest butterfly systems down there, and very much in trouble from fire.
Okay.
But it could be one that citizens could have a lot of input in, and probably one of our most imperiled butterflies in the country, and one of the coolest and prettiest. It's called the tailed copper.
Tailed copper.
Yeah, check it
Very
out.
cool. Definitely.
You had a lot of support and interest for the Pieris Project early on, and some funding. Are you still able to get grants to continue your work, or is it, how is that?
I could. I just I don't know if it's final, but I'll be getting adjunct status at University of Tennessee, Knoxville. I'm mentoring a student there, and that's where my dual appointment was University of Tennessee, Knoxville, and NC State, and so. Yeah, so as an adjunct, I think I can apply for a grant still. I haven't done that yet. We still have so much data just to sift through. So that project I showed you, like what other DNA is there? We don't even need to spend any more money. It's just time. And so a lot of what I'm doing to further answer the question, Robert, about time is like I'm trying to offload some of these things to other
Yeah.
professors or grad students. Like, hey, if you can use this, use it.
Yeah.
Here's this great data set, or if you want to collaborate on this. But things, yeah. The thing is that I only showed you some of the things I worked on. I was just very ambitious, working on lots of different things. There's other really cool projects related to phenology, has nothing to do with butterflies, just phenology more broadly. And that's like one of my most interested papers I'm most interested in. And I just slowly moving that along. But
Sean, I'm sorry, I'm talking too much, but I gotta say one more thing. I think the advertisement for this talk, there was a pejorative used about the cabbage. I don't remember what it was. The cabbage white, the small white, Pieris rapae. But I have to tell you, among a lot of us, it's a very much beloved butterfly because it's often the only butterfly we have around. Yes, 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 Carolyn. Because I mean, I'm speaking to you from Astoria, Oregon right now, and it's the only butterfly that's commonly seen in Astoria, and it's seen from March to October, and people love it. And I dedicated my PhD thesis to the cabbage white because it was the only butterfly I was seeing during a lot of that work. So we love that butterfly, and thank you very much for bringing it more alive for us.
yeah, and you're not alone. I met another student, actually maybe two or three now, that were trying to work on different butterfly systems, and it just wasn't working. So I think one is the West Virginia white,
Yes.
and she was having trouble, so she switched to most of her work being with the small cabbage white. I think that's what ends up happening. It is a great lab. I mean, it's a great species to study because it's everywhere. And people have been studying it. Actually, there's a cool paper on like the angles of the wings and how that could be used for photovoltaics and things because it intensifies the solar radiation or something. It's just they've been studied in physiology, so there's a lot of work. Joel Kingsolver's lab, UNC, that study them, and it's just they're a great system. And I don't want everyone to collect all of them. The thing is, I don't think anyone's gonna decimate this species. It's robust. But it's kind of like it's a great ambassador species, and to the point of what I said earlier is that like to connect people. Now people are like, oh yeah, small cabbage white butterfly instead of a cabbage moth or I have no idea I didn't even notice it, right? It's funny how you kind of live like blind to things going on in the world until you made that connection and then you start to see it. It's like when I worked for the USDA as a fun summer job. That wasn't mentioned in the beginning, but the most fun job I've ever had was working for the National Forest Service as an undergrad, where I went out looking for spotted owl and goshawk nests and spending the summer out there looking for nests. It's like when I left, now I see them everywhere at the corner of my eye. It's just like it's like a superpower. And I think that getting more people to be aware and just be observing of what's going on is awesome. And that's one of the benefits of citizen science. So I think, sorry, I'm talking a lot too. Like a lot of people say, well, if citizen science, if someone else can do it, like with Zooniverse, there's an argument. Well, if by the creator of it, if people, if like machine learning or an algorithm can do this, then why have citizen science? This is for the things that the machines can't do. But I would argue that's not necessarily a good way to think about it, because there's other benefits other than generating science to citizen science, like generating data or even analyzing data. It's connecting people with, the scientific method or the system they're working on.
And the natural world.
Yeah, the natural world. So if it's something outdoors, like iNaturalist or other things, you're getting people to pay attention. iNaturalist is great now. They'll like guess what species it is. Hopefully all of you are familiar with iNaturalist, but yeah, you can basically just take a picture and it's pretty dang good. It'll guess what species you just photographed. And so that helps people connect even further with what they're seeing and generate useful data.
So any other comments, questions, thoughts? I actually have a question about the. I mean, we're all critter lovers, and so, but looking at it from the other side of the coin, are the cabbage whites still such a scourge for the agricultural industry?
No, they're not on the top 10, 20, 50 list probably anymore because BT is so effective. And we kind of speculate in our paper or just raise the idea like, well, maybe there's something with this loss of diversity into why they can't overcome BT. Because the diamondback moth and the cabbage looper, two moth species that are also agricultural pests of brassicaceous crops, those have overcome. They've evolved resistance. Yeah, the cabbage looper in the lab and the diamondback moth in the wild. Diamondback moth is the first to develop resistance to BT. BT is the Bacillus thuringiensis, this like bacteria that produces this product that it basically attaches in the gut of the insects and makes them starve to death. And so it's very effective against the small cabbage white butterfly. You spray some of this on. It's an organic, so if you eat organic, that's one of the tools that's used because it doesn't kill many other species. So they have ones that are like more focused on certain species. But yeah, anyways, it's very effective. And so at the moment, unless they evolve resistance, the farmers can treat them, but it costs them money. So if you ask farmers if they could snap their fingers and get rid of the small cabbage white butterfly, they would all do it. And like I was saying with the in New Zealand, the large whites, they were working very hard to keep that out for similar reasons. Is that they can be devastating. Or you just have to pay a lot of money to keep up with them. You can do it. You just have to pay it.
Pieris brassicae, which appeared in Brazil in the 90s, I think. Here's a little story about Pieris rapae in England in the 1960s, I think. Jack Moriarty published on this, but cabbage whites were or small whites as they call them there, were an enormous pest on Brussels sprouts, particularly in the county of Bedfordshire. And so they used increasing amounts of DDT for it. This is pre-Rachel, pre-Saint Rachel. And it was effective for a while. And then it became less and less effective. And they had to use more and more and more. And what Jack Moriarty at Monkswood was able to show was that the cabbage whites had evolved resistance to DDT, but the carabid beetles, which were their primary predators, had not. And so the carabid beetles were knocked off, therefore increasing the survivorship of the cabbage whites, which were resistant. And so the system became more and more simplified. The cabbage whites just went from strength to strength. They're extraordinarily adept organisms. That's one thing I love them for, Carolyn, is that they're not just beautiful, but they're smart butterflies. I'm convinced they're quite clever butterflies. And they are extremely well adapted, and they do what they're evolved to do extraordinarily well.
Yeah, for sure. I mean, I was just gonna say they have to be. They're so successful, right? Sorry, go ahead, Jonathan.
Houseflies and rats. I mean, they're all like really cool.
For sure. Yeah, I think there would be a great citizen science project with a housefly, right? Everyone knows them. They could collect them. So the DDT, so this was in Europe? This was in England or?
Yeah, I can send you the literature if you'd like. It was done at the Monkswood Experimentals. No, I'm sorry. It was done at Rothamsted, the great entomological research station in Britain in the 60s.
In Britain, yeah.
Yeah,
they're very diverse over there, genetically diverse. They're very genetically diverse there. So I wouldn't be surprised. Here, or like Australia, they're the least diverse, right?
Right, they're almost homozygote.
So there they'd be less likely. But I still imagine, and if we ever finish that project, to looking at adaptation, like contemporary adaptations, I imagine that they are evolving, adapting. Maybe not, they've lost some, ability to overcome things like maybe DDT or other things, but when it comes to like adapting to different climates, I'm pretty sure that their critical photoperiods, so the time at which they decide to wrap things up, is probably different at
I
different
was
latitudes.
extremely interested that you got a measurable diversification from the railroad transplants to California, the pink dots. Tell us more about the pink dots. How different were they? How much, what did you find in terms of differentiation there?
Yeah, they're quite different. Like, how would I? Yeah, I don't have a slide to show you like a three-dimensional genetic space, but they're quite distinct. And that's just because they had, a couple decades to
Couple decades, yeah.
couple decades of a very small group of individuals that are probably very genetically similar to grow into this population that was large enough to just basically, and if you think of the geography too, right, you have the mountain range that is a natural barrier, so they're not going to be really coming, there's not going to be many coming over that mountain to try to mix and kind of dilute or homogenize between east and west.
Right.
So that barrier, and then there's the northern barrier, somewhat of a barrier, and so yeah, that's what kept them from distinct long enough to where they're kind of impermeable to mixing.
Very cool.
Yeah, but there is mixing, like I said, there is some evidence of gene flow, like in Washington and other areas, where the genetic ancestry of the ones in the West are found up in the Northeast. And I didn't mention, which I should, because you guys are in Washington, that Joel Kingsolver, when I first showed him some of this stuff, this was very preliminary, didn't have any of the cool slides I showed you guys, but I had done the genetic structure analysis, and he was interested in Washington because they had done some lab studies where they collected them in Washington and compared them to North Carolina to looking at what I was telling you, the seasonal adaptations. And so getting to the point here, he showed that they're different, that they're different in their development. And so he was asking, are they genetically distinct? And what I was saying earlier is that if you look for like within North America or another, like in Asia, you can find more genetically distinct if you look at a finer resolution and kind of look a little bit harder. And the Northwest is somewhat distinct. So the butterflies in Seattle are actually fairly distinct from the ones in the rest of Eastern US, but there's still enough alike that they could be grouped for our analysis. But so you have a distinct population up there.
You know, Joel used to be here at the UW.
Yeah.
So there's a question, Sean, about predators. What eats the cabbage white, and is it geographically different across the United States?
We do, right? I just almost had one tonight. Yeah. So the farmers aren't completely successful. You find the little black gooey things in your broccoli sometimes, but yeah. So one of the students that I had a picture of, Davel, who's Colorado State, Paul Ode was his supervisor. They were looking at Cotesia, which is a parasitoid. So that we actually brought over some predators. So they didn't have a lot of predators here, or at least not there aren't that many documented predators. Birds will eat them, but in terms of other insects and like a dominant, like really good at preying on them predator, there aren't many. And so we introduced these two parasitoids, or basically like wasps, that will lay eggs in the larva of one of these, and then they'll develop and emerge alive. It's like, kind of where they got the idea of aliens. So they'll come out of the caterpillar in these little pupal sacs, and that's one predator that we have introduced, but it hasn't been very successful. Or I should say two at least, and it might have even been a third parasitoid, but those are the two. There was Cotesia and is it rubella or something? I can't remember the names of these wasps.
Presumably those were introduced by the agricultural industry, or?
Yeah, they were introduced early on, so I don't, maybe early 1900s. I can't remember when they were introduced. This is the same as they introduced them in Australia, I believe, and I'm not sure about New Zealand. I think New Zealand as well. It was just it was a parasitoid in the native range, and that's kind of it's a biocontrol. So that's a common approach, or has been. Well, we'll introduce something. And so with the fire ants, there's the forward fly. So we've tried to do that where people have developed, rearing these forward flies and they'll lay eggs in the ants' heads and decapitate them. But they just don't really put a dent in them. I mean, it's just once they've established and they're in the millions, there's just no stopping them.
Yeah,
That's
as I say,
interesting.
in England, they did find that one pretty effective predator was carabid beetles on the larvae. And they're not that commonly preyed upon by birds because, as Remington showed, they are aposematic. They're distasteful because of the mustard glucosinolates that they sequester in their tissues. And Remington thought that Charles Remington at Yale thought that the entire group of Pierids, all the whites, were one big Muellerian mimicry complex because none of them taste particularly good. They're not as unpalatable as monarchs, but none of them taste very good because of the mustard glucosinolates. So yeah, no, they don't have a whole heck of a lot of predation on them, I don't think.
Yeah, I knew that about the caterpillars. So it's the butterflies as well too, huh?
Yeah.
Well, if you think about the butterflies, they fly slow, they flap around, and they're white. It's not like they're not obvious in their environments, right? So you could say that's an aposematic, coloration, right?
Right, because you find individuals with bird strikes on the wings, but there's not that much observation of actual predation. It's a gradient. If a bird's really hungry, it'll probably eat one.
Yeah.
It's not gonna probably barf like a barfing blue jay.
Same is true with the caterpillar, right? Yeah.
Exactly.
Anything. Yeah, I did some. It was funny because I did some studies in my master's. I was at Bowling Green State University in Ohio, and I was doing these preference trials with. That was the diamondback moth and the cabbage looper, and giving them. We were using these transgenic Arabidopsis. So we were basically playing with the glucosinolate chemistry, and then looking at which ones they preferred. And my advisor was saying too, it's like, we were talking about, well, the experimental design, and it's like, well, if they're starved, it's like they'll eat anything, it's just like, yeah. So exactly. It really depends on what's around for those predators, and it's probably a number of factors.
Well, it's interesting that there are predators in England, the carabid beetle, but not so much here. The predators had to be introduced, so maybe it's just a question of time.
Yeah, and I don't know if anyone knows or has looked at, maybe Robert would know, the natural predation rates. I just don't think anyone's really looked at it that well, and it could have changed over the last 160 years they've been here. Probably the predators are probably feeding on them more than they used to, one would think. But
yeah, probably so. It's probably facultative, is probably opportunistic. One of the most important carabids that feed on them in England is the big greenish purple refractive iridescent beetle. I forget its name. John, do you know its name? It's that.
I think it is a carabus, actually, or
It's a
Caponota.
Carabid. I can't remember the genus, but people probably see them in the gardens. It's quite a big. It's almost 2/3 of an inch long, and a bit of a produced head. Beautiful purple green reflection.
Oh, yeah, that is a carabus.
Also, they're introduced from Europe. Also, I'm sure they're feeding on them.
You know what else I think?
The genus is carabus.
Yeah, that's right. It's a genus carabus, right?
I want to say, and I haven't confirmed this, but having raised grown a bunch of cabbage and kale in the backyard to do some experiments with my daughter raising caterpillars, I noticed the lizards were hanging around. So these are western fence lizards hanging around, and it looked like they were picking off caterpillars off the plants. So that would be one predator.
Yeah. I always tell people just to steam the caterpillars with your kale, with your broccoli, You get a little protein that way. It's a way for a vegan to get a little protein.
Yeah, it just looks so awful when they're this black goo. Yeah. So another one of the, so I should say, there is a virus, a baculovirus that can kill them, and that's part of some research I was going to do prior to leaving academia through the USDA, was to look at variation in these baculoviruses. And so basically, it's a virus that they crystallize and they'll be dormant on the plant material and in the soil, and when the caterpillars eat them, they come to life again essentially, and start replicating, and they turn the caterpillar into this black ooze that then just drips on the other leaves, and then the other caterpillars will eat that ooze, and then they'll die. And so that's a biocontrol, but I think in terms of scalability, they're very effective, and they're very, very host specific. So even better than BT, I think, in terms of like just killing, a small group of species. So just like, maybe pyrids, or even maybe just even, the genus. But in terms of scalability and mass production, I think that's where it breaks down.
Yeah, it should be said that if people want to use BT in their gardens, I recommend not. It's not very hard to just pick off the larvae from your crucifers.
Small scale operations. Yeah,
that's right. Because if you use BT in your garden, BT is not specific. So you're going to kill all the moths and butterflies in your garden if you introduce BT in your garden.
You can just net. You can just put those nets over your plants.
Yeah.
And that will keep them from ovipositing. Or you can get out there with your net and collect them for a project, and then they won't lay anything in your garden.
Very good.
But, yeah, there was some guy in Australia that would shoot him with a BB gun, apparently. He said, "Do you still want them?" I was like, "Sure, there's still DNA in there.
Yeah.
If you're going to shoot them, I might as well take them." But
sure.
So, Richard, I see you smiling. It must have been okay to have a whole presentation on the little cabbage white.
Well, I just think this was a great idea for a program, Paulette. And thank you, Sean. This has really been interesting. For some reason in my yard, and maybe for all of us, it seems like we've just had a cabbage white summer. Just really not a whole lot of anything else. We had some Western tiger, but it's been a cabbage white summer. So this is perfect timing. Yeah. So well done, Paulette. Thanks. And thank you, Sean.
Oh, you're welcome. Thanks for having me.
Thank you.
Okay.
Yeah, thanks, man.
Comments, questions?
Nice to see everybody.
Okay.
I have a question for Bob Pyle.
Yes, ma'am.
So how's the Dark Divide doing?
Who's speaking? Is that Kim?
It's Carolyn.
Oh, I'm Carolyn. Where? Oh, there you are.
Yeah, here I am.
Oh, the movie?
Yeah.
It's doing very well. It's getting wonderful reviews. It's November 10th. It goes into regular streaming platforms, everything except Netflix, and it'll be in DVD. I recommend the DVD because it's going to have a lot of deleted scenes. Sean, I'm sorry. It's
can you bring Sean up to speak about what you're talking about?
The David Cross film, right?
That's right. He plays me.
Yeah.
It's nuts. It's nuts. Your talk is much more interesting. But the
so it's supposed to be based on you?
Yeah.
Oh wow, I didn't know that. That's
David
more
Cross
fun.
Plays me, man. It's weird.
Except the guy in the movie is really bumbling, and I don't.
Well, this is the
Bob's
thing.
Bumbling,
You
but
know,
it makes an interesting movie.
Everybody loves the movie except my best friends because they think they're protective of me because he's such a doofus going into the woods. I'm actually a pretty decent woodsman, or at least I was. But the thing is, in a narrative, it's like science. You've got to have a story. You have to have development. You have to grow. You have to discover. And if David went into the woods as a good woodsman, where's the story, So I'm happy to take one for the team. I go in there as kind of a dolt, and I come out a little bit better. And it's not totally out of character, as Pelham will tell you. So, yeah, it was a lot of fun. But the great thing is that Deborah Messing plays my late wife, Tia, and she's absolutely stunning. It's a good movie. I recommend it to everybody. And Sean, I'm sorry to get an advertisement at your expense, but it's
Oh, no.
Carolyn's
I-
fault.
I saw the trailer because I went on your website to look at some of the things you had on there, and I saw that you guys were excited about that. And I was
Yeah.
like, oh, that sounds fun. And I know David Cross from Arrested Development, so
Yeah.
it's like,
He's
I watched it.
remarkable. It's his first full-length feature role, and it's a strong dramatic role for him. But that's the other thing. He had to have some room for his comic chops, right? And he gets some at my expense, so that's cool. I'm laughing too. Thank you,
That's
Caroline.
awesome.
for asking.
It'll be more exciting watching it now that I'm knowing that's based on you.
Yeah, well, I've been watching you for an hour and really enjoying it, so I hope to give you something back for that. Thank you.
Okay, any other comments, questions?
Thank you very much, Paulette, for doing this.
Okay, happy to do it. Thank you so much for joining us.
Hey, and remember our happy song.
Happy
I might not be speaking for everybody, but just remember our happy song tonight. Ding dong, the witch is dead. Witch, oh witch, the wicked witch. Bye-bye.
Thanks, everybody. That was great.
Okay, thank you so much for joining us, Sean, and we may have you back to talk about swallowtails in the future.
Yeah, definitely. Yeah, I was gonna say too, if you guys, I don't know how often you pick people, but Roger Villa, who's in Spain, especially now that everything is web-based, I think people are more open to doing things, and I can talk to him. But yeah, he's quite the butterfly scientist. He's remarkable work. But yeah, I can think of some other people too. But
Okay, I may hit you up.
wonderful.
Okay, thanks so much, Sean, for joining us tonight. You have a good evening.
You too.
Thanks, everybody. See you
Take care,
next
everyone.
Bye.
Okay, bye-bye.
Cheers.